Hi folks, I'm Anusha, currently pursuing civil engineering degree. As a part of quarantine activity, our professor has assigned us a new work. In this video, I will be presenting a seminar on aerobic bacteria, anaerobic bacteria, pathogens and non-pathogenic organisms, which will be based on the subject water supply and treatment engineering. So let us get into the basics. So the basics get into the definitions. First of all, in every topic, or whenever we talk about the basics, first thing that come to our mind is of definitions. So let us go through these definitions, simple definitions. These are one sentence definitions. First, aerobic bacteria. Aerobic bacteria, as I am showing, those which require free oxygen for survival. So as the definition says, the bacteria which can survive or which can complete their life cycle in the presence of oxygen, they are known as aerobic bacteria. So a small animation for you. Consider this as an oxygen molecule and the bacteria is happy. It is surviving in the presence of oxygen. So we can come from that. We can say that the organisms that can survive in the presence of oxygen are called an aerobic bacteria. Once the oxygen goes from the bacteria or once it is separated from the bacteria, the organism dies. It fails to survive in the medium or in the environment. Such bacteria are called aerobic bacteria. So the second one is anaerobic bacteria. Those which flourish and thrive in the absence of oxygen. It means the organisms which can survive and which can complete their life cycle even in the absence of oxygen. Such organisms are known as anaerobic organisms. See, if consider this as an oxygen molecule and this organism is surviving even in the absence of oxygen, even from the separation. So such kind of bacteria are known as anaerobic bacteria. Then third one is pathogenic organisms. Pathogenic organisms, the organisms that spread harmful diseases to humans and animals by entering their tissues and causing serious waterborne diseases. Such organisms spread through contaminated water, food or anything. So what basically pathogenic organisms is? The organisms from which various diseases spread to humans or animals are known as pathogenic organisms. Such organisms are very harmful to either humans and animals also. So the last one is non-pathogenic organisms. So what the definition says, bacteria that are harmless under certain conditions and are beneficial to humans, animals and plants are called non-pathogens. So there are also organisms, not all organisms or microorganisms are harmful to a human or animals or plants. There are certain organisms that are beneficial, those are harmless. Such organisms are called pathogenic organisms. And even in certain conditions or in favorable conditions, such organisms are beneficial to humans, organisms and animals. Such organisms are called non-pathogenic organisms. They are harmless. So guys, we gone through the definitions. Now let us consider some colorful images so that we can understand easily. So this is the assignment and the topics covered here are aerobic, anaerobic, pathogenic and non-pathogenic organisms. So the first example for aerobic bacteria is Escherichia coli, which is most commonly called as E. coli. So these are harmless aerobic microorganisms which are found and residing in intestines of all warm-blooded animals, including human beings, and are thus excreted with the feces. Since these harmless organisms of coliform group live longer in the water than pathogenic bacteria, it is generally presumed that the water will be safe and free from pathogenic bacteria if no coliform bacteria are present in it. Although many bacteria are useful and helpful in cleaning natural raw waters, certain bacteria often found in water produce fatal diseases. Whereas the usual routine tests are generally conducted to detect the presence of coliforms, which in themselves are harmless organisms, but their presence or absence indicate the presence or absence of pathogenic bacteria. So what it means is, basically E. coli is harmless, but we can conduct certain tests which is known as membrane filter technique, with the help of which we will be able to decide whether the water is contaminated or not, or whether it is contaminated with pathogenic bacteria or not. So the E. coli is basically found in human intestine. These are certain symptoms: low grade fever, vomiting, stomach cramps, and diarrhea. So this is what I was talking about, the membrane filter technique. This is the simplest and most recent method adopted for detecting and measuring the presence of coliform bacteria. Is to filter water sample through a sterile membrane of a special design. The porosity of this medium is of 5 to 10 micrometer. So on which the bacteria will be retained after passing of water. The membrane is then put in contact with nutrients basically known as M-endose medium, that will permit the growth of only coliform colonies types. So after an incubation period of about 20 hours, here we can see the incubation period for 24 to 48 hours, the coliform organisms are developed into visible colonies which can be easily counted. So this method is called membrane filter technique. Now let us go through pathogens and non-pathogenic organisms. As I have already told, most bacteria are harmless, and under certain favorable conditions, they are beneficial to human beings. Such bacteria are known as non-pathogenic bacteria, or commonly non-pathogens. However, certain other bacteria are deadly foes of man and animals and may enter their tissues, causing serious waterborne diseases such as cholera, typhoid, infectious hepatitis, etc. Such harmful bacteria or organisms are known as pathogenic bacteria or pathogens. As we have seen here, the pathogens spread from one person to another person by mutual contact and other things. So the diseases caused by pathogens are harmful to human beings, animals. So these are some examples of pathogens. Some of these cause diarrhea, hepatitis, typhoid, and certain other infectious diseases. These are some other examples of pathogens. So guys, I think you are clear with the topics aerobic bacteria, anaerobic bacteria, pathogens and non-pathogens. If you have any queries regarding these topics, you can comment in the comment section. At last, I would like to thank Professor Asha Avika for giving such a wonderful opportunity to present our seminar. In these quarantine days, she helped us in spending some quality time. Thank you, ma'am, and thank you, friends.